So I'm so excited to have you all here. My name is Pia Sorensen. I'd like to welcome you this evening to our Science and Cooking Public Lecture Series. This is our 10th year. We're celebrating a 10-year anniversary. So very soon, I'm going to introduce you to our speakers for tonight. But first, we have some business to attend to. So who was here last week? Oh, nice, nice. So then you were here, and for those of you who were not here, Joanne Chang from Flour Bakery was here. She taught us all about the science of sugar, and she boiled sugar to many different temperatures. And so who's here for the first time? Anyone? Okay, good. Welcome. This is awesome. So then I have to explain. So we do this thing every lecture where we play a game. So if you went to the class last week, and if you paid very close attention, then we ask you a few questions about last week. And if you know the answer, you raise your hand. You don't say anything. And then you have the chance to win one of our aprons. And for those of you who've been here before, you know what we do when we see these aprons. So to translate, when we see equations, we clap. That's what that was about. Okay, so ready for the question. Let's do this. So Joanne boiled sugar to different stages. Which stage contains more sugar, hard crack or firm ball? Raise your hand if you know the answer. Yes. Yes, that's correct, hard crack. Now, the question is, why? Oh, so that was easier. How about you?
Because more of the water's been removed at that point.
Why has the water been removed?
Because it's been boiled off.
It's been boiled off, and left behind is the sugar. Do you know how much sugar is left behind at the hard crack stage? You get the apron anyway. Oh, wild guess, anyone? 97? I
think so, 99.
99. That's correct.
They had the numbers.
Yeah, maybe. Oh. The problem is we're going to run out of aprons. Do you know how much sugar there was in the firm ball stage? Anyone? I think that's like 87. So it goes from 87% sugar to like 99. Yeah. Okay. Third question. Joanne made praline. What sugar stage is that? Raise your hand, don't say anything. Anyone? Yes? Caramelization, correct. Okay, we're going to have to ship this up to the gentleman in the middle back. Okay. So fourth question today. So what do these people have in common? Yes. They all do fermentation. That's correct, but it's not all the way correct. Anyone else? Yes. Yes. They've all been speakers in this class. That's correct. So usually at this point in the semester when we get to the fermentation class, usually I show a picture of all kinds of different foods, and then I ask you, so what do all these foods have in common? And you all kind of snicker, and you're like, fermentation, because that's where you were here. And that's correct, but it turns out, and I thought of this yesterday, all of these people kind of cover the diversity of all of those foods. So Tara Whitsitt and Sander Katz both came and talked about sauerkraut, lactic acid fermentations. Last year, Gabriel Bremer and Michael Harlan-Turkel talked about vinegar, fermented food. Jody Adams a few years ago talked about pickles, yogurt, and bread. Sister Noella Marcelino, also known as the cheese nun, talked about microbes and cheese, also fermented food. David Chang, a long time ago, I think this is back in like 2013, talked about his pork bougie, which was his version of katsuobushi. It's basically fermented pork, which he sort of discovered by mistake in a corner in his kitchen. Andoni Aduriz talked about the beautiful creations he makes, sort of coating little apples and other things with various molds. So they kind of look inedible, but they're actually beautiful and they're delicious. So all of these foods, and many, many more, are all fermented foods. And so usually, after I sort of showed you the slides, so all the foods, and I said, okay, what do they have in common? I would then go on, and we would marvel at how diverse they are. And then I would go on and I would tell you that the reason microbes are such amazing cooks is because they divide so quickly. You very, very quickly go from just one microbe to many, many, many microbes. I would go on to tell you that in the process of doing this, they create preservatives and all kinds of amazing flavor molecules, right? Does that sound familiar if you've been before? Who has been to any of these? Yeah, nice, so a good number of you. Nice. Do you remember these things, right? And then we talked about how the key with cooking with microbes is all about how you cook with life, right? Because you need to keep these little creatures alive so they do the cooking for you. And I thought today, I'm not going to go through all that, because this time I'm sort of joined by two very science-y fermenters. And this really, introducing these two people to me, is really sort of bringing all of this full circle. Through all this time, for the sort of 10 years that we've been doing this, the Noma Fermentation Lab, sometimes coupled with the Nordic Food Lab, has been sort of leading the fermentation revolution, has been sort of inspired all of these people, and now, finally, they're here. So David Zilber, you may know, he's the director of fermentation at Noma. He came out with this book with René Redzepi last year. I was super excited when this happened, because I was like, finally, I have a textbook to use in my class. Finally. Jason White was formerly at Noma, and he's now the head of R&D at Kudzu, which is Sean Brock's new restaurant in Nashville. And I'm going to cut this short. Let's go ahead and welcome David and Jason.
Things are just too easy in the Kavango Basin. They no longer have a zest for living like their country cousins out there in the inhospitable desert. Here, life offers no challenge, and many of them have become slobs and easy riders. The only excitement in their lives comes in high summer when the marula trees are in full fruit. At first, the marulas are very tasty and hard to get at. You have to be either agile or strong to get your share. But after a few weeks, the marulas get overripe and they start dropping from the trees. Then all the animals wade in and gorge themselves on the rotting fruit. It starts fermenting in their tummies and turns into a very potent brew. So on the way home, things start happening to them.
Hi, everyone. I'm Dave. P introduced me, but I just like to say that. And I'm here to talk about fermentation with my homeboy, Jason.
Yo, what's up?
That was a little video that Renee had me find for the book tour when we launched this book. It is called Animals Are Beautiful People. It was produced in 1974, and it was an exploration of the African savanna that happened to have a very hilarious little detour into the world of fermentation. But I know that fermentation is a very trendy topic these days, but as you can see, it predates any cognitive sense in any animals around ever, because it's been happening for basically all of history. And the proof that animals gorging themselves on alcoholic fruit and having a good time of it in the African sunshine is no exception to that. But it is a nice way of illustrating that fermentation is all around us in every sort of way. And it is absolutely a part of the natural world and really inescapable. And it's really fun when you get to cook with it and actually put it to good use. But to start off, I know that now I was warned in advance that you guys are probably better educated than the class that's going to be here tomorrow night. But I always start off with just the question, what is fermentation? What is it? Just shout it out. What is it? Microbes? Not all microbes, but they're involved. What else? Yes. Yes, so yeah, take those two answers, smash them together, and you get fermentation. I would say, and it is a very colloquial but very easy and useful way of talking about fermentation. I would say that fermentation is the transformation of one food into another by way of a microbe. That's the best way to look at it. Pia might have a much more technical definition of it. No, it's fine, it works. Chemists and biology students might say, oh, it's an anaerobic enzymatic pathway that yeast uses to metabolize glucose and transform that into ethanol in the absence of oxygen. Yes, that is the technical chemical definition of it, but it doesn't go far enough, and it doesn't help an average Joe understand what fermentation actually is or what it does or how you use it in your everyday life or what it means to the world of food or civilization or humanity at all. So it's much easier to just say, there's a bunch of life you can't see, microbes. They take some foods and they turn them into other foods. And that's the point, because that's at least a little easier to understand. Now, if you want to really get into it, you do have to talk about human intent. And humans have a big part to play in fermentation, because without the human willing fermentation into existence, you just have a free-for-all. And there's an analogy that I use in the book, and it's that as a fermenter, you're taking the role of a bouncer outside of a nightclub somewhere in downtown Boston, all the Celtics fans pouring out of the bar after, looking for booty or whatever. And they're lining up outside, some drunker than others, some happy to be there. You as the bouncer, you have to decide, okay, who's going to come inside and start a bar fight, and who's actually going to come inside and order drinks and have a good time and, be gregarious and enjoy themselves and actually make the bar a nice place to hang out for all the other customers. So as a fermenter, you're deciding which microbes to let into the club. That club being all the food you're looking to ferment, the ingredients you're looking to transform, and which microbes to keep out. Good guys and bad guys. But that delineation, that distinction between good guys and bad guys, that is strictly human intent. Life, all life, for the first 2.5 billion years of life on Earth, it was all microbial. And microbes don't have any sort of morality. They don't care whether the microbes next to them are thriving or had a good day or a bad day. They care about what's out there, what's around them. They can sense things. Some scientists argue that the first sense to evolve was in fact taste, because the quorum sensing that microbes use to actually figure out what's in their environment, that's all just chemical communication. And what is taste or smell if not chemical communication in between the outside world and a living organism? So microbes, they're just trying to live. They're just trying to get out there in the world, divide and multiply, be fruitful. Some of them put up porcupine spikes. Some of them put up defenses to make sure that the microbes next to them don't invade their space. Now again, these are just happy accidents of evolution in nature, nature finding clever ways to turn some things into other useful products for those microbes to do their thing. So when you talk about fermentation and you talk about even the classical sense, ethanol. Ethanol is a poison. Anyone who's been out a little bit too late on a Saturday night knows this on a Sunday morning, but it does mess you up. And it also messes up other microbes in an environment of yeast. So if yeast is consuming sugar and producing ethanol, it's not to get you drunk. It's to keep its environment safe for it and it alone. So the creation of exclusionary chemicals in a fermentative process is at the heart of it all. Now, botulism produces botulism toxin. It's a microbe. It produces an exclusionary chemical, and it's an exclusionary chemical that is no good for anyone in this room. A very small amount will kill you. But lactic acid bacteria produce lactic acid. Now, lactic acid is something that how many people had yogurt today? Maybe for breakfast? Yeah, there you go. Y'all like lactic acid. So you can see that there's obviously a difference in between what sort of exclusionary chemicals are produced in the world of fermentation. And that's an important distinction to make, that there are going to be some chemicals that are actually tolerable to you and some that will drop you dead. Now that delineation is exactly what we're after. That's the thing that we want to get to the heart of in this talk and understand, all right, who's in our favor and who's against it? Because that's all fermentation is. It's just deciding. It's just being the shepherd, deciding where your microbes are going to wander in the world. It's about being the farmer and deciding what to plant and when and what season, and saying, what do I want to get out of this and what do I want to keep out? To plants, a field is a field and a plant is a plant. It's only a weed if you choose to pull it out. But it's a crop if you put it there and wanted to harvest it at the end. And that's exactly how you have to think about fermentation. Yeah. That's the intro. All right, so we have microbes. And for starters, we're going to talk about lactic acid bacteria. They're kind of the poster boy. They're the easiest ones to get your head around and use. There's some very simple ones that are homofermentative bacteria that really just take sugar and turn it into one byproduct, which is lactic acid. Now, that's not the whole story. There's a lot of other things that go on when it comes to bacteria. But for the most part, we're going to just, for ease of use and ease of thinking about it, going to lump them all into one category called lactobacilles. Now, I was going through old videos of Ferran doing this, and he had really fancy slideshows, and I don't know, that's not very Noma, and Richard Feynman didn't use PowerPoint presentations, so I'm not going to either. Now, lumping bacteria, forgive me if there's anyone who knows these things better than I do, and this is not an accurate depiction, but it'll suffice for now. Lactobacilles, a group of bacteria that have the characteristic of producing lactic acid. Now, taxonomically speaking, that's a terrible way to describe them. Any Cladis in the room is going to be like, David, that's bullshit. But it's a very useful way to talk about what these things do. Now, talking about lactobacilles and all of that huge swath of bacteria that just happen to do one thing is a bit like talking about all animals with 4 legs. Yeah, you can jump them into one category, but you're also going to dump in alligators, elephants, dogs, insects that lost 2 legs into the same category, but you're actually going to have a useful description. Now, it's especially useful because we're going to talk about the products that it makes and how that affects everything you want to do with them. So lactobacilles come across glucose molecules. I think it's like a hexagon, but we'll just call it a G. They use their enzymes to break down those glucose molecules, extract the chemical energy, build ATP, or adenosine triphosphate with it, power all the things their body needs to do, and in doing so, they leave behind that exclusionary chemical we're talking about, which is for every one molecule of glucose, it is 2 molecules of lactate. Now, evolution is wild, and it is amazing that nature can come up with all of these clever ways to do one thing and do two things in the same shot. So yes, when your body consumes glucose, does it produce lactic acid? I don't know. Is that what like cellular respiration, like muscle burning? Yeah, it does. It does. Okay. But I only have a high school diploma, so forgive me. But it is also fascinating that these bacteria have evolved to specialize in doing this, and they do it extremely well. And not only are they specialized in producing lactic acid, but they're also specialized in a way that they can tolerate it. They can actually survive in extremely low pH environments. And so when a lactic acid bacteria is thriving in your fruit, and let's say that this is botulism. I'm going to give it devil horns because it's evil. And it starts producing lactic acid. It will now outcompete the botulism because as the botulism bacteria comes into contact with that lactate, well, the pH is a little bit too low for it. It oxidizes and dies. So it's really about clearing the field for that bacteria to thrive and grow, and not for bacteria like botulism to thrive and grow. Now, you're saying, okay, well, I can add lactic acid bacteria. I can get the powder. I can use a bit of backslop, you could say. If you've ever made yogurt at home, you just take a tablespoon of the old stuff, put it into fresh milk, let it sit on your counter for a day or two, and bada boom, you have yogurt. That's because you've introduced that population. But it's also important to note that you want to give, if you're trying to be a successful fermenter, you want to give something like lactic acid bacteria every opportunity they have, every chance at their disposal to edge out the competition. You want to take the active role as a fermenter to go the extra mile and basically drop your kids off at school and say, okay, you're going to get there safe. So as a fermenter, you have certain tools at your disposal to be able to do this. When it comes to lactic acid bacteria, we have, well, let's just call them control points. We have access to oxygen. We have salt concentration. We have pH.
Food.
We have food, yeah, a nutrient source. And also temperature. That's 5. There are more if you get into the nitty-gritty, but this is a good start for all the basic things that you would see on this table. So when you're trying to cultivate lactobacillus in a product of food, whether that's shredded cabbage for your sauerkraut, whether that is the plums that you see on this table, you're trying to build an environment for that microbe to survive. And that's exactly, like I talked about, you want to be the shepherd. You don't want to take your sheep out to pasture in the middle of winter and let them graze through the snow and freeze to death. No, you want to do that in summer. So it is all about these small things that you can do as a fermenter to make sure that the things that you're trying to ferment actually have that leg up. Lactobacillus have certain types of things that work for them better than others. It's just like if you were to take a penguin out of Antarctica and put it into the Amazon rainforest, it might overheat and die. But likewise, you couldn't take a bird of paradise out of the Amazon, transfer it to Antarctica, and expect to have a blooming flock in a year or two. All life is conditioned and built around the environments that they grow up in. The environments in the organism are not two parts of different things. They are the same thing, just on different facets. So when you talk about lactobacillus, you have to say, okay, well, what does this bacteria really need to thrive? That is an environment without oxygen. There are some that can thrive in the presence of oxygen, but for the most part, lactobacillus are anaerobic bacteria. And another thing to note is that they're totally fine and happy to propagate in the existence of a salty environment. They are halophiles. Halophiles meaning salt-loving. So if you have that knowledge at your disposal, you can add salt, and you can remove the oxygen. Let's kick that out. And all of a sudden, you have an environment that the lactic acid bacteria is more than happy to thrive in, but the botulism, less so. Now, botulism, if you know, is also also an anaerobic bacteria. It's also happy to thrive in the absence of oxygen, but it has a lower water activity, a higher water activity level than lactobacillus do. And that's something really important to know. Water activity is the amount of free water available in whatever medium you're talking about, whether that's the human body, a bowl of fresh peas that you're looking to ferment, a vat of shredded cabbage. And it's about how much available water is around for any life process to really take hold. A vat of cabbage with no salt added has a water activity level of 100. You add 2% salt, that drops a little further. And all life on Earth has some threshold where it's going to have a lot of trouble getting like a foothold and actually kind of kicking things off. Botulism, like I said, has a higher water activity level than lactobacillus do. So by adding salt, you're actually slowing it down and giving the lactobacillus a little bit of extra room to do its thing. And that's exactly what you're doing while you're fermenting. Removing the oxygen, again, it's moot for these two, but things like molds can't take hold in an environment that's oxygen-free. So again, you're cutting off at the pass any malevolent microbes that you might think might grow in the presence of all of these things. And then from there, the lactobacillus feasts on all the glucose that you've produced, or that you've given it to do its thing. The lactic acid's produced. As the lactic acid's produced, the pH of the medium, whether that's, like I said, an urn of sauerkraut or a bag of fruit, drops, and then there's no chance after a certain amount of time that any of this stuff can actually take hold. And that's how humans discovered fermentation, by realizing that it was a pathway to safe food preservation. This goes back thousands and thousands of years. I mean, the first records of people fermenting foods go back at least 10,000 years. I think there was discoveries in like near Iran of like people making flatbreads and actually leavening things and making sourdoughs there. But the first hardcore evidence that shows up is about 6,500 years old of urns of wine that show up in the Zagros Mountains. Now again,
like Sandra Katz said, though, like that was really just the first record of pottery. You know what I mean? Like it goes back even farther for as far as we know, honestly.
Yeah. You can't assume that the first time something shows up, in a physical object was the first time it was ever made. So it does go back much farther than that. But like I said, it is an amazing means of food preservation. And it must have happened by accident. Like you see, fruit falling from trees, ending up on the African savanna floor, is a reminder that these things are processes that have always happened and will always continue to happen. So somewhere in our ancestors' deep history, they must have happened on a process and just realized that, yes, there's two things at your disposal. You have your foods that you were looking to keep. Someone added salt to it, a known mineral that you could find and were maybe looking for. Human settlements often made camp at ocean seaside, so that would make sense that would be around. And just by the sheer understanding that, okay, the thing that I had with salt water lasted longer than the thing that I had without it, you would just naturally keep doing that process. There's a famous ethnologist that experimented with pigeons, and he made them do all sorts of songs and dances to get fed. It's kind of the same thing with humans in fermentation. We had no understanding as a species of how these processes actually worked. The entire field of microbiology is only a little bit more than 125 years old. But still, people have been doing these things for millennia. So it is important to understand that beyond just drying food, the first real food technology we had for safe food preservation before the USDA was a thing was fermentation. And it makes sense that we do it until this day and that we have all these traditional recipes on our countertops because these are things that have kept civilizations alive for a very, very, very long time. And we've grown up to actually love the taste of them and understand them as a part of our cultural history. So I digress. It is, I think the best way to talk about fermentation is as a form of symbiosis in between the living world and the humans within it. You think about all of the amazing research and leaps and bounds that have been done in human microbiome research these days over the past, 10 or 20 years, that we're finally beginning to understand, almost like a Copernican revolution again, that no, the world is a lot larger than we ever thought it was. Now, with the discovery of the human genome and understanding how life actually forms in the last century, this is all amazing stuff, but to understand just how much microbes play into our life is another step further. The fact that microbes like yeast or lactobacillus became friends with us, became basically domesticated by us like wolves did and ended up being turned into dogs is something that I find extremely powerful and amazing to think about. But these microbes exist by the droves. I mean, if you scrubbed all the life off of Earth, all of the things that you would consider to be living things, from plants and grass and plains to trees and dogs, animals, whales, it doesn't matter, and all the humans, like the world would still be teeming with life. It is no short understatement to talk about, the world being a microbial world, especially when you consider the amount of time that life has existed on this planet and the amount of time that multicellular life has existed. Even if you're to extrapolate the history of the Earth into the future and watch the sun grow to a red giant, after it got too hot for anything big to survive, there would still be microbes. So microbes are really life, and even multicellular organisms are just a strange collection of smaller single-celled things. The symbiosis and this amazing kind of interplay in between humans and the microbial world, though, is something that I find extremely powerful, as I said. You have this unseen world that works to your favor if you just know how to treat it right. And in humans, we find partners in microorganisms from yeast to lactic acid bacteria to acetic acid bacteria. And in us, they find an environment as well. And this is something that I find profound about fermentation that really borders on philosophy. And it's not just that, yes, these are tools, and yes, these are things you can look up in a microscope and name and catalog, but organisms are environments to each other. Organisms can become entire playgrounds and entire ways of living for each other. All the microbes that serve humanity also find us pretty useful in ourselves. Yeast, Saccharomyces cerevisiae, it's amazing. It gives you beer, it gives you wine, it gives you bread, but we also give it the ability to thrive and reproduce. And what is life if not the attempt to thrive and reproduce? So it is kind of powerful in that sense. And in the act of working with these microbes and eating them, and again, talking about the importance of really breakthrough human microbiome research, we can understand that the boundaries in between the outside world and the inside world, what we would consider ourselves as humans, as like walking organisms, and if everyone has an idea of themselves. But it's hard to understand yourself if you don't understand that you're also teeming with tens of trillions of microbes in your gut and on your skin and in your eyebrows and in your armpits and behind your ears and in your mouth. They are a huge part of you. It is an amazing idea that you are almost on equal footing, cell for cell, with as many microbial cells as you are human cells. And that's a lot of foreign DNA to carry around inside you. But again, you are a vessel, an environment for these microbes, and they help to make the environment of you one that carries on healthily. Doing research for the book in its writing and talking with actual fermenters from places like the Caucasus Mountains in Georgia, you realize that traditional societies that have done these things, fermented, used lactobacillus to keep milk safe for weeks to feed themselves, actually end up with an amazing kind of mirror image of the microbes in their environments inside of themselves. And yes, you can get your gut tested. Yes, you can send your poo off to a lab somewhere in California to get everything analyzed and have a diet crafted especially for you. But the relationship, the human relationship to microbes isn't that, superficial. It's not like you could ever take one magic pill and say, okay, I'm going to fix myself by eating a little bit of fermented food. Like I said, in all my research and talking to fermenters from Georgia, the most impressive kind of feats of this microbiotic symbiosis come from cultures that do these things as a way of life. They do these things not just to subsist, but to thrive. It is as much like a part of their existence as anything else. As much a part of their existence as it might be for any Harvard student to show up at class every day. It's something that is practiced and passed down via tradition through families and generations, across continents, so on and so forth. And I think that there is something extremely poetic that, biology uses the term culture for a petri dish full of microbes, but humans also use the term culture for tradition and history and storytelling and craft and recipes. There is this kind of beautiful link there that I just can't get over. And to quote Jane Bennett, who wrote on materiality, I was sent this quote by an architect friend of mine, but she digresses from, the state of the natural world and all the things you could use to fill your life, buildings and objects and things like that. And she talks about food for a second. And she says that the act of eating and the consuming of materials deemed dead, but still in fact teeming with bacterial life, is that of the holobiont, or the blurring of boundaries between the inside and outside of human bodies. And it is very true. A holobiont comes from the Greek whole and organism. And that is the idea that, yes, you are not just the human genome and a species, but you are all of the collection of all of the phenotypes that you could ever possibly encounter in your life. And that's always changing as well. From the moment you're born, you're blessed with all of the microbes that your mother had with you in her breast milk and in her vaginal passage. And that's your first kind of christening into the microbial world from the amniotic sac. And then from there, all of the foods that you eat, all the microbes in your surroundings, all the microbes in your environment, all the microbes that produce all of the foods that you consume end up as part of you as well. And they, all through your life history, contribute to your existence as a holobiont or a whole organism through your life cycle. And it's amorphous. It's changing. It's an ecosystem, and ecosystems are never actually stable. They're always in flux. And so are you. And that's why it's an amazing thing to understand fermentation on this deep level and understand that life is this constantly changing thing. Both Jason and I know the struggles of working in a 2 Michelin star restaurant that strives to be the best in the world and using the tactics of fermentation to transform food in really impressive ways that wows people, that impresses our boss and excites Renee with something genuinely new. And then also know that, okay, well, the Michelin Guide values consistency. That's a tricky situation.
Yeah.
Because life doesn't give a damn. It will do what it wants to do. But that's also a beautiful thought to surrender to. We'll talk about all this stuff on the table in a second, but I just wanted to get through that little poetic moment. Okay, so, and maybe that is a perfect segue to talk about what we do at Noma and why we use fermentation there at the restaurant at all. If we could switch to the next slideshow. This little houseboat is something called the Nordic Food Lab. But we're going to have to rewind from this being parked outside of Restaurant Noma in Copenhagen a little bit and talk about Noma's birth at all. Noma was a restaurant that was started in about 2003 by Renee under the mandate that we would have, or that there would be a Nordic restaurant that kind of focused on and concentrated on Nordic food, or the food of the region. Now that includes Denmark and Sweden and Norway, all of Scandinavia, up into the Faroe Islands, and Greenland, Iceland. It was less a geographical box and more of a cultural box, more of a historical box of all the places that the Norse might have gone in their history over the past 2,000 years and explored, camped out, and made settlements. But there was an impetus to kind of link this cultural history and this cultural heritage and distill it into a restaurant. Because up until the early 2000s, fine dining food in Scandinavia was French food. And fine dining ingredients in Scandinavia were imported ingredients, giving it short growing season. So Rene came along under the impression that he would, buy into this Nordic manifesto that a few other chefs in the city had put together, and they would focus on cooking, or defining, rather, the food of a region using what they had at their disposal. So Rene went and did that. He opened Noma in the dead of winter in 2003. Again, like I said, with the idea that he would use, the wilderness and everything that grew in the region, only to find that he'd opened and nothing was growing. A bit of a mistake of timing. But that did give him and his team of just 8 cooks at the time the ability to really dive into the cultural heritage of the region and do a lot of just research. And they looked through old Swedish army texts that would teach you how to forage in the wild and teach you what foods you could eat for a whole year if you needed to. They started contacting foragers in the region who would bring back some, wild goods, whatever they had cellared, until one day a man named Roland Rittman, big, portly dude, looks a bit like Santa Claus, hard of hearing, drives a big rickety van, and always rolls up to the back of the restaurant, even to this day, with goodies and supplies that he forages from the Swedish forest. And he brought out a bag of ramson capers. And Rene was like, this is amazing. These are the buds of wild garlic. Here in North America, it's Allium triticum, which is ramps. And in Europe, there's a cousin called Allium ursinum, bear's garlic, or wild ramsons. That grows a little differently, but they are very similar flavors. In the late spring, it produces these tiny little seed pods that Swedes have traditionally turned into capers for a very long time. They're amazing little punctures of garlicky, sour, delicious flavor. Rene was kind of captivated with this idea that you could forage for goods and preserve them. And they started doing that with gusto come spring. They went out to the beach for one of the first times as soon as the first shoots started popping up in early March. And Rene, trudging through the ocean seaside, in their galoshes, spots a little patch of green growing out of all of the rotting seaweed, walks over to it, picks it up, just smells it, and he's like, this could be edible. Snaps it in half, chews on it, and he gets the taste of coriander. This was beach coriander. Now, you guys all know coriander. There's Thai restaurants and Vietnamese places and Mexican places all over the city. But to a Dane, back then, in 2003, coriander was a very exotic flavor. And it was quite surprising to him that there would be this exotic flavor in a very cold and dreary place like Denmark. So they started cooking, like I said, with gusto, with all these wild plants that they now went out and foraged for, just the cooks themselves before coming into work every single day. One day there was wild horseradish on the menu. Rene had picked some, they were using it on a plate, and he'd ask one of the foragers that would work part-time and just bring back different goods and wild greens to pick some for him as well. The next day, the horseradish is in, they put it on the plate, and it has 0 spice. And Rene freaks out. He's like, the spice was making that dish, and now it's not there. He calls up the forager and says, why doesn't the horseradish taste the same as it did yesterday? The one that I picked was spicy, and it was full of flavor, and it was alive. He's like, well, Rene, there's 150 different types of wild horseradish that grow in Scandinavia. You didn't specify which one. And Rene was like, really? Enter the houseboat. Rene, realizing just the sheer amount of knowledge that needed to be written down and documented if he was to really understand how to cook with the wilderness in a restaurant setting, in a fine dining setting, teamed up with Copenhagen University and launched the Nordic Food Lab as a purely academic project that was actually kind of sneakily a Noma project. But their mandate was to kind of catalog the edibility of the natural world. And that's exactly what they did. At first, it was to catalog the edibility of wild plants in the wilderness. And they did that, and they worked very hard at it. It was researchers, academic students, PhDs, and master's students, and 2 cooks from the restaurant as well, and that was kind of the interface between the 2 entities. That there would be this joint cooperative project. The cooks would work alongside the researchers and actually figure out what to do with the foods, and that research would feed into the restaurant as recipes. After they started working through the idea of getting all these wild plants, preservation came back up, and Rene remembered these Ramson capers and said, okay, well, if there's Ramson capers, what else can we make capers from? Making capers is a very simple process. The standard caperberry bush is a bud that blossoms in the spring. You take it, you pick it, you pack it in salt, and that's a caper. Add it to your puttanesca, amazing. But Rene, wanting to translate these ideas to the Nordic setting, to Nordic ingredients, they started out on the caper project. What can we find? What unripe berries are out there that kind of fit that same mold as the caperberry? How can we pack that in salt and keep it for the winter months so that we don't suffer the same disaster of flavorlessness that we did the first year we opened? And so the chefs in there, Soren and Torsten, and all the researchers from the Nordic Food Lab started messing around with salting. And that's all they thought it was. They thought it was salting. They thought preservation was equal to salting. They did it with gooseberries. They did it with the Ramson capers. They did it with unripe black currants. But of course, it had to sit and mature. Months and months later, Torsten reaches down into the fridge and pulls out an inflated bag that looked a little bit like this, but more puffed up, of gooseberries. Rene walks in, and Torsten's beaming, and he says, "Chef, taste this." And it's a murky liquid. And the bag looks a little strange. There's like some white goo in there. And Rene's like, "What? What's this?" And he's like, "Just taste it." Rene tastes it, and it's like fireworks. It's not just salty berry juice. This is something else. This is complex. This is lactic acid mingling with the phenolic acids from the berry. This is salt, but also layers of umami. It's tart, it's sweet, it's sour. It's all the flavors in one little flavor bomb, and they immediately start cooking with it, bathing oysters in the juice, slicing it thin, putting it over fresh fish that they would wrap in papillote with all the wild herbs like goosefoot and groundnut that they could find. And the restaurant was indeed transformed with one ingredient. A little investigation, they tracked back through the records and they realized after reading the label, oh, this was a messed up experiment. Someone didn't weigh out the salt correctly. They didn't add 10% salt, they added 2%. And then it was just left to sit. But it wasn't bad, it wasn't rotten. They didn't taste it and spit it out. It tasted good. So what had happened? Well, the research team got on it and they discovered that, hey, they had fermented. It was a very happy accident. And from there, a Pandora's box was opened. They said, okay, well, yeah, this is the same lactic acid that grows in sauerkraut. So if we just made a sauerkraut of berries, what else can we make sauerkraut from? Can we make sauerkraut from plums? Can we make sauerkraut from, what have we made sauerkraut from?
Leaves, fig leaves.
Fig leaves.
Everything you could think of.
What else?
So we made it from kohlrabi.
Cherries.
We made it from cherries. We made it from celeriac.
Peaches.
Peaches.
Apricots.
Strawberries.
Strawberries. Green strawberries.
Green strawberries.
This is going to be a long list.
Herbs.
Yep.
You know?
Anyway,
Yeah,
it is.
It keeps going on and on and on.
It
I mean,
does.
where there's a food stores for lactic acid bacteria and you have some water and salt, I mean, really what you're remembering about the actual sauerkraut itself can be reminiscent in many things, so many things that have a food source for that bacteria. It's like almost limitless.
Yeah.
It'll drive you wild at night thinking, what is this going to taste like?
He has this problem. But you're exactly, no, he's exactly right. It was a discovery that wasn't just like, okay, we made something new, this is cool, let's use it. It was a discovery of possibility. They discovered fermentation not as just a recipe, but as a tool to be applied to anything they wanted to. And thus, that Pandora's box. So they discovered that they started fermenting, and by this point, a man named Lars Williams quits at the Fat Duck and joins the team in the Nordic Food Lab. He was my old boss. And Rene, now that they're going buck wild with all sorts of lacto-fermented fruits and vegetables, sliced thin, celery, kohlrabi, broccoli stems, berries, cherries, black currants, white currants, using these ingredients on their menu, he tasks Lars and says, okay, just dive into the world of fermentation. What is it? What else is out there? What products are fermented that we don't understand or know about? And how can we use it? Rene, having done some trips to Japan and understanding that they do have this deep culture of fermentation, tasks Lars with the creation of miso. But again, through that same lens of applicability of one process to another. What grows in Denmark? Not soybeans and not rice, as goes the traditional recipe, but yellow peas and barley. And very soon after knocking on the door of Japanese ferments, Lars inevitably discovers koji. And koji is fermentation's, well, at least fermentation at Noma's, but really in the whole world of fermentation, one of your best friends. Koji is Aspergillus oryzae. I got space over here. It is a filamentous fungus that got its start as a blight, as a crop pest that used to spread through Southeast Asia. It likes humid climates, and it likes warm weather. Anybody here renting like a really cheap and kind of shitty bachelor apartment with black mold growing in the bathroom? I have at some points in my life. It's nothing to be ashamed of. That black mold that's causing you asthma, whether you admit it or not, is a relative of Aspergillus. It's not Aspergillus oryzae. It is Aspergillus niger. Aspergillus niger produces something called aflatoxins. These are toxins that are the same exclusionary chemicals that we talk about when we talk about things like lactate. As the mold lands on whatever food source it can find, whether that's wet PDF fiberboard or food like onions in your cellar or a vat of grains, it will do its thing. The spores land and they start to release their hyphae. This is what a mold spore looks like under a microscope. Actually, we have one here. Can we switch to that feed for a second? There we go. So this is also a black mold. And there you see its spores. Those tiny little buds. It looks like a dandelion. There's its stem, tracing all the way back to the grain. Oh, there's more of the cluster to that organic matter that you see there. Just a little bit more. Focus, focus. Ah, not so good. Anyway, you get the idea. So that's an Aspergillus spore, or Candida. And that's what it looks like after it's been grown for about 72 hours. But at the start, just one of those little specks, one of those little balls, should it have all the right conditions conducive to its environment. Basically, a humid climate, warm weather, and a nutrient source. Let's call that a grain. It will begin to release its enzymes and multiply and spread its hyphae. Hyphae are the tendrils that many a botanist up until the 1970s thought was an analogy for, not even an analogy, thought was another form of root like you find in plants. It is crazy to like look at the history of science in retrospect and be like, they didn't know anything. But that's not how science would, like, yeah, scientists don't know anything until they do. But as late as 1974, the entire world of fungi was considered a branch of Plantae. It was not its own separate kingdom. We now understand it as such, and we now understand that, yes, fungi are eukaryotes more closely related to mammals than they are to plants, even though they function very differently. But it is a fascinating world to dive into. So as this spore starts releasing enzymes, and enzymes are the name, we'll talk about enzymes in a second, starts releasing enzymes and garnering useful chemical energy for its own cells, it spreads those filamentous hyphae and grows its web. After it's grown enough and consumed enough of its resources, it'll take all the sugar that's produced by seeping enzymes into its nutrient source and chopping up things like starches or cellulose into useful smaller molecules, and it will sprout a spore. I'll draw that over here. and basically enter its reproductive cycle. The wind comes along, and all these spores, like dandelion flowers, go off into the wind and continue that life cycle again. Be fruitful and multiply. Aspergillus niger, in this stage, to make sure no one else is messing with it, like I said, produces aflatoxins that are poisonous and that will irritate you and make sure that you stay away from whatever it's trying to eat itself. Some lucky bastard or brave soul must have also realized that when this pest grows on food, it also transforms it and makes it sweet. That's because the enzymes that it produces break down things like starches, which are long chains of simple sugars that are bound up in such a way as to not be useful to an organism unless you have a way of breaking it up. Now, when you eat starchy foods, if you chew on like a saltine cracker long enough, you'll find that like the mash in your mouth will go from being quite bland to actually a little bit sweet tasting. And that's because your saliva has the same enzymes that koji produces in it in small quantities, because that's the first point of digestion. Any organism that tries to take in a little bit of the natural world and transform it into useful energy has to break it down in some way first. The production of enzymes is how this is mediated in all life everywhere. Enzymes are proteins that have the unique ability to be at once keys and scissors in a weird way. They're keys in that they fit specific locks, and scissors in that they can actually chop things up in very precise kind of cuts. The enzymes that the Aspergillus family produces are multiple. I won't talk about, sorry, I was about to get really sidetracked and talk about laundry detergent. But the enzymes that Aspergillus produces, the ones that we're concerned about are, again, we're going to talk about classes. There's amylases that break starch up into sugars, and proteases that break chains of proteins up into their constituent molecules. And those are the ones that we're really concerned about when it comes to fermentation in this realm. Enzymes seep out of the organism into its base product and cut these molecules up into that free-floating glucose that even the lactic acid bacteria was so concerned about. Sugar is nature's battery pack. It is prevalent everywhere. The transformation of carbon from just floating in the air or the ground into useful molecules of like packed chemical energy, like glucose molecules, at the hands of plants through photosynthesis, is one of nature's truly great feats. And it's no wonder that all life on Earth in some way tries to seek it out and get its hands on it. Once you have those glucose molecules, the organism can do whatever it pleases with it, move motor proteins through its body, break down other molecules. It is really like that AA battery in the natural world. But in this state, it also tastes really good to us. And that's why we seek it out in the first place. So, getting back to that ancient culinarian who might have happened on this very special process, he tasted it and started cooking with it and realized that this mold was actually extremely useful and then started cultivating it. And that was kind of that big leap in the world of fermentation. All these things, like I talked about with that vat of cabbage that was left unattended or unsalted and the one that wasn't, and actually cared for or understood that, okay, I did something that made this one way and not another. These are these like interesting forks in the road of human history where an accident led to two different outcomes and one was preferred over the other. The discovery that Aspergillus could transform foods into things that were either more nutritious or tasty or useful was another such moment. So, an Aspergillus strain ends up being cultivated, and slowly but surely people realize that, okay, if I breed this thing, if I keep throwing it through generations, and I have a few nifty tricks at my disposal, I can kind of select for a strain of Aspergillus that doesn't make me sick, that doesn't make me cough. And that's exactly what Japanese fermenters did, going back almost 2,000 years. The Japanese All of this happened in mainland China at the start, and North Korea as well. But when it made the trip to Japan on the back of Buddhist monks, they really got otaku with it, which is Japanese for very nerdy. And koji, being the national mold of Japan, is really something that is cherished because it is responsible for basically this entire amazing empire and nation's cuisine. It is the basis of things like amazake that they would eat in the morning, of the soy sauce that they could have with almost every meal, of the misos that they have for miso soup,
Beer,
cook with,
spirits,
for beers, for the spirits that they drink, for
porridge, just the
vinegar
porridge.
that they season their sushi rice with.
Absolutely.
It is really the backbone of that whole, half of that continent's culinary underpinning, if you will. So they domesticated this like, wolves were domesticated from, or dogs were domesticated and transformed from wolves. Traits that were beneficial were kept, and traits that were malignant were discarded. And those lines were then perpetuated and bred up until the present day. And there is incredible diversity in the world of koji. You can see here that just visually, this is just 3 samples of just maybe the dozen or so that we have at Restaurant Noma. But they can be green, they can be black, they can be tawny, they can be yellow, they can be stark white, like we have on the corner over there. And this is all just through the sheer variation of just artificial selection, or people deciding what something's going to look like in the end and selecting for different traits. This is Aspergillus oryzae, but different from the one that we have on the corner in that it's green. This is Aspergillus awamori, which looks like the black mold that I was talking about earlier, but is actually completely safe to eat. And this is Aspergillus luchuensis, which has the nifty ability of producing citric acid. So in that kind of big world, you discover all of these different avenues for the creation of different food products and the creation of different flavors. Jason is going to show you how this actually goes down in the restaurant or at home if you're a brave fermenter. And he's got all the tools ready.
Yeah, so I mean, this isn't exactly how I would do it probably if I was in the lab, because one, I have 3 different kinds of spores just hanging out together, and so that probably wouldn't be the best practice if you wanted to grow a specific strain. And whenever we're growing koji at Noma or at home or in a business, what we're trying to really do is try to get a specific type of koji that we like and really just nourish it and harness its abilities in incubation and make sure that it's not infected with yeast or the things on your hands. Sure, you can mix koji with your hand, and that could be fine. But if we're working in a place like Noma, we really have to make sure that the product that we're starting with is what the guest receives as well. So we don't want to have all of our hand bacteria on our koji necessarily. So it's very important that we really just cherish the process and follow all the science possible to ensure that this wild process can be as contained as possible. And essentially, just like cooking, we use our senses, whenever we want to know when the barley is ready to cook, to actually grow mold on, the way that we tell, we could do a water test and we could find out the water content of the grain through research and also just through texture and taste. I'm like, hey, Dave, is this barley good? And then he says,
barley's good.
So then I say, so as a cook, you really can kind of stop thinking about the science and all the stuff that can be overwhelming for a minute, and you can just be like, this is ready or not. But it's very important. We have a standard kind of recipe at Noma. It's 100 degrees Celsius for 45 minutes with grains that have soaked overnight. It's very important for them to become saturated with water so that as it's in incubation, it can, 1, produce a little bit of its own humidity, and 2, also give the fungus the opportunity to grow within the grain. So once the barley has been cooked and steamed and mixed a bit to kind of make the process of cooling expedited a bit, we let it cool down to room temperature, right? Because if you were to just get these spores that I have here and throw them on hot barley, it would be like throwing a baby in a boiling pot of water. It would be disastrous. So,
Would.
Exactly.
It'd be disastrous, but less illegal.
Yes, less illegal. So One of the first things that happened to me when I first started making koji is like I kind of just winged it. I just winged it, you know. I was making koji in a barn, and I was just like learning from different videos and people and traditions that were passed down through my friends' families. And a lot of my koji just died straight away because I wouldn't have the patience and the time to kind of like let it come to like body temperature, essentially. So once it comes down to temperature, you just put it inside of a pan. You wouldn't want to just put the whole entire bowl inside of this pan. If you did that, it would just be so many grains in such a small area that as the fungus started growing and kind of like encompassing the grains themselves and producing their own energy, it would just overheat and then start fermenting while it was growing fungus. And then when that happens, you don't have a stable batch. And the sugars can actually become sour, and it can spoil and get really weird and slimy. So for this pan, like I'm kind of just filling it halfway, maybe a little bit more, you know, just so that way it has enough surface area to where it's not crowded, right? And once you have the barley inside of there, we at Noma, it's really important for us again because we don't want wild koji growing everywhere, like different batches that taste like something completely new every single time we make one strain of koji. Typically we use lab purchase spores. Well, we have good companies that we work with. We get the spores from them, and we use it directly from the package every time. That being said, David and a lot of his colleagues in the past have explored harvesting your own spores from spore-related koji batches. It works, you can do it, but the more that you grow it outside of a laboratory environment, the more opportunity you have for it to get infected, for other things like yeast and foreign molds and bacteria to become part of the spores that you're in turn going to be adding to your grains, and then eventually it becomes something else. One of the people from the Nordic Food Lab, Josh Evans, is actually working with us recently on the evolution of Aspergillus, growing it batch after batch after batch from the same spores. Once you get the spores in the canister on grains, you just kind of like let the seeds kind of like fall. This is a lot of spores. You don't need to do that much spores, but for the sake of entertainment, I put on. Honestly though, like you could add the tiniest bit of spores and put this in the right environment and it would grow beautiful koji. It's a misconception that you kind of need to add so much like extra flour or extra spores in order to make the koji grow better. You want to make the koji grow well, honor the process. That's what you need to do.
A tablespoon of the pure powder, like a tablespoon of the pure powder that Jason was shaking out of that canister, contains 1 billion spores. And that's a lot.
Yes.
Just a few of those spores, just a few hundred, would be able to colonize this pan of barley and grow, given the right environment, and spread. And like every spore is that organism starting a life cycle anew. So to think that you're putting 1 billion organisms into this pan of barley in the effort to make something like this is a lot to think about. I mean, in the wild, as a wild crop fungus, a wind carrying just one spore off to a new field would be enough to cause a blight a few months down the line. So when you're using it to ferment, it is important to realize that, yes, there is a lot going into this thing. Now, another thing that's really important to realize when we're talking about koji, and especially when we're talking about domesticated Aspergillus, is that if you were talking about Aspergillus niger, yes, there are exclusionary chemicals that give it a leg up over its competition. If you're talking about Lactobacillus, yes, it produces exclusionary chemicals that give it a leg up over its competition. If you're talking about an organism that has been bred to be completely benign, what defenses do you have against anything else growing in its stay? Effectively, none. Other than that, of colonization resistance. And when we talk about colonization resistance, we're talking about adding so many spores that it's going to win the game no matter what. Going back, I'm from Toronto. So imagine the Raptors and the Golden Gate Warriors coming up against each other, but it's only Curry on the San Francisco side and the whole team of Raptors on the other. It would have been done in 4 games flat, right? And that's colonization resistance. If you give the microbes that you are looking to work with such an initial population that they're just going to outcompete everything else that might be around it, even if it's there in a plentiful supply, you've ensured that that microbe's going to win and your koji's going to work out just fine.
Yeah, absolutely.
So
Yeah, so once you have this little package of koji, it's really important also that you don't have a dry towel, and also that you don't let your koji dry out before it even goes into the incubator, right? Like if you have a wet towel, it actually helps with the humidity becoming in contact with the grains as they begin their incubation. And then also it helps with it creating that environment of humidity on its own. If you make koji in a proper environments, you don't even have to set your humidity from the beginning. But if you have a large batch of koji in a large room, then it does help to have your humidity regulated from the beginning. But some producers don't even use humidity for the first half a day. And also producers in Japan, and they don't risk having infection either. A lot of them use pure spores as well. So once you have everything ready to go, you got a damp towel and you got your koji, you put it inside of the incubator. We do our koji pretty standard at Noma. It's 30 degrees Celsius, roughly, and 75% humidity. We do make a lot of koji at Noma, though, and a lot of kind of like specific spaces that need a lot of attention. We never just put something inside of the incubator and we're like, we know what we're doing, it's just gonna work out. Though we do know what we're doing, we're constantly monitoring these things. We're seeing that this tray is hotter than this tray. The humidity in this room is slightly different than this room, and then we change our stuff around. We never just think that things are gonna work out. What we really try to do is make sure that we are in touch with the process and we're in tune with the way these things are developing and leaving our knowledge like on the table and helping this organism function properly. Yeah. That's really what it's all about.
It is about crafting those environments. In the same way that talking about lactobacillus, the kind of easiest one to wrap our heads around, you're looking for the environment that it's happiest to grow in. When you talk about koji, you're trying to craft that ancestral field somewhere in southern China, where in the summer, it's 30 degrees out, it's humid and sticky, and there's a panda walking by somewhere in the corner of your eye. But that's exactly what it is. When Noma travels a lot, we've done pop-ups in Tokyo, in Sydney, and in Tulum in Mexico. And as a fermenter for 2 of those, myself, I had the most fun in Mexico because it was the ancestral environment that koji grew up in southern China. All I had to do was exactly what Jason did, and then I just left it outside. Why? Because it was 30 degrees all the time, and the moist air blowing in from the Caribbean Sea made it a beautifully humid environment. And the koji grew amazingly because it was the environment it needed. As a fermenter, it's all about crafting the environments that your microbes need to thrive and leaving the microbes that you don't want out of the picture by ensuring that those environments are conducive to the microbes you want and them alone. That's koji.
Yeah, absolutely.
That's a good walkthrough there. But it is amazing. You guys got samples, or they're coming. In the bottom cup is a little piece of what we have on the corner of the table here, which is our very standard albino variant of Aspergillus oryzae grown on barley. So kind of rewinding the tape a little bit and going back to fermentation at Noma and the history of how it all started, Lars was tasked with making miso, and he happened upon koji because it is basically 50% of a miso. All the enzymes it produces, when it's grown on something like rice or a grain like buckwheat or even barley, seep into that grain and break up those starches into sugars. But like I said, that's not the only thing koji produces. It also produces proteases. Proteases have the amazing ability of breaking up proteins into their constituent amino acids. Like I said, keys and scissors. So again, forgive me if there's like a biotech student in the classroom or something, but I'm just going to draw a protein. It might be steel wool. You'll never know. But as koji's enzymes come into contact with a protein, they'll start snipping it off through hydrolysis, using the water in the environment to cleave apart these molecules, again, into their constituent building blocks. And those little amino acids that are left behind are then freely floating in whatever medium you're looking to actually produce them in. Now, in a grain, you find not just starch. Let's say that this is a grain of barley, which koji is just as happy to grow on as it is rice. Little anatomy class here. We have the husk, or the pericarp. We have the starch, or the endosperm. And then we have the embryo itself. But there's also a fair bit of protein in grains like rice or barley. And koji, being the nifty little creature that it is specialized in getting all the energy it can out of those too. So that's where koji's proteases come in. This is the protein coat. Now, the best analogy I've ever heard about what a grain or a seed actually is comes from Thor Hansen, who's an American naturalist and biologist. And he talks about seeds as being basically a mother plant's baby is sent off to school with its first lunch wrapped in a lunchbox. The husk, that's the lunchbox. The starch, that's the lunch. And the baby's sitting right here waiting to get into it. Now, the mom isn't stupid. The mom knows that there's a lot of kids out there that would bully its child and want to steal its lunch on its way to school before it can start its day. So how does the mom prevent that from happening? Well, like I said, it binds things like starch, or actually the useful chemical energy inside of it, in long molecules that need a special key to get into. For larger physical threats, you have the husk, which keeps out larger organisms. But for things like other microbes, if you don't have the enzymatic keys to be able to break up starch or protein, you can't do anything with it. Now, the embryo itself is equipped with enzymes that are actually capable of breaking up the starch and protein. And those are the same amylases that koji produces. Now, it's interesting that koji produces these. The naturally equipped keys that a mother supplies its own baby plant because, well, it's a parasite. That is what koji is. That is what the Aspergillus family is. It's a crop parasite. It's an organism that's evolved to mimic all of the biological processes that the plant would use to get at its own most valuable resource. It does that through the same protein complexes that the plant produces itself. Now, when the embryo has its perfect set of conditions, when it's landed in soil, when it's perfectly moist, and when the temperature is just right, its biological processes kickstart. It starts releasing its amylases into the lunchbox itself, thus breaking up those starches into sugars. And it'll actually cut up those amino acids into proteins as well and use those to build its bodies, or its cotyledons, those first shoots that you see on a baby plant. They're kind of pale yellow, and they're kind of droopy before it can actually photosynthesize. That's what that lunchbox was for, those first few days of life before it can actually make its own food itself via the sun. This process that I've just described here, the first early days of life of a plant, is something that everyone enjoys on the 4th of July. A cold beer. It's malting. That's what this is. When maltsters are looking to make their wort and make a beer, they get that baby plant, they trick it into starting its life cycle for only just a couple of days, then kill it by roasting it, and then using all of that now degraded starch, that is maltose, or a simple sugar, and brewing that into their wort, which is then fermentable by yeast. If you were to make a porridge of grains like barley or rice and just leave it to ferment, it wouldn't turn into beer. It would turn into gross, bad-tasting, foul, off-tasting slop. But when you actually employ these enzymes to transform a grain or to transform its base processes or its base products into something else, you get something that's actually fermentable by a whole other class of organisms, like even things like lactobacillus. Yeast, Gluconoacidobacter, all sorts of different varieties of life. Now, in Asia, they never happened upon this. They had to find koji, and they had to go through this whole process of domestication to get to the same point where they could take their staple crop, their grains, rice, and transform them into something that was fermentable by yeast to make everything like sake, to sochu, and amazakes, things like that. It is kind of just an accident of history that the West didn't discover aspergillus and that the East didn't discover malting, but so it goes. Now, the cool thing is that you could never make a miso with malt. The baby plant doesn't produce enough proteases to be able to use them in the same way that you use amylase to harvest that sugar. But with koji, you can. So by doing the same thing with koji as maltsters do with malt, you grow the organism just short of its life cycle. You stop it from actually going into its full-born reproductive stage and producing the thing that we saw underneath the microscope. You grow it for 2 days or 48 hours instead of the 72 that would be required to actually get it to produce spores. Now, why is that? Has anyone ever tasted like a leafy green like lettuce or arugula late in the season after it's gone to seed? It's bitter. That's because the plant is using all of the sugars that exist in the plant's body and putting that energy into the production of that next generation of cells. The same thing happens with koji. At the second day, it's sweet, it's fruity, like what you just tasted. It's full of flavor. It's not just boiled barley, it's a transformed product. But if you were to let it grow for another 24 or 36 hours thereafter, that sugar would be used up as it starts producing all of its Candida spores. So by cutting that life cycle short, you now have this little packet, this amazing chemical toolbox that you can add to other ingredients to transform them. And that's exactly what Lars happened upon when he made miso for the first time. He took barley koji, using something that grows in Scandinavia, using that base product, translating an idea or a process from somewhere else, looking to tradition to figure out how to basically transcribe flavors or ideas, umami and sweetness and complexity to things that grew where we were. Jason's going to show you how to make miso right now, and we're going to talk about all the important factors that go into actually producing one successfully so that it looks like this and not like a batch of moldy food.
Exactly. With the miso that you guys just tried, it started with ingredients like this. You know, it's like it's crazy how such a dense and hard ingredient can become broken down into something that's so sweet and umami rich, and that's the beauty of koji. You know, it's almost like the perfect like marriage of its protease and amylase activities. And besides that, like when we're making koji, for instance, like we're trying to put it in an environment that's really safe, and we don't want all of this outside bacteria. We definitely don't want our miso running rampant, but one of the beautiful things about the miso process is that we allow it to breathe. And that way, whatever yeast are in your environment that are salt tolerant, that live alongside your miso, actually contribute to the flavor. So in a way, we want to make sure that we do it very precisely. So we have our ingredients at the right temperature. We also have them ground so that we can increase the surface area. That is very important because koji is powerful, but if we just throw the beans and the koji inside of a jar without doing anything to it, it's going to have a lot of extra oxygen. It's going to have a lot of little extra headspace scattered out throughout it. And then on top of that, the food that the koji would otherwise be consuming is actually difficult for the aspergillus to digest. So that's why we increase the surface area. And by doing that, we simply grind it by hand or inside of a meat grinder. We have a pretty standard ratio that we do at Noma that's definitely inspired by ancient practices, roughly 60-40 and 4% salt. David can explain a little bit of the heartaches of 4% salt, I think, if he wants to.
Yeah.
But it's really untraditional to make miso at 4% salt. I mean, if you make miso at 4% salt with the normal water activity that you would have in a standard miso recipe, you're going to have a lactobacillus party.
That's very
at best.
That's very true.
So a lot of that has to do with just, again, using your senses as a chef. Really, at the end of the day, like I know what a miso is supposed to feel like at 10 and 15 and 20% salt. So when I want to start mimicking that texture, right, what I'm trying to do really is make sure that it's not too wet. So one thing I can really tell whenever I'm mixing this miso, for instance, is that when I squeeze it through my fingers, it's not just shooting out all wild because it's so wet. There's not so much water in here that it's like a soupy mess. It's very dry and dense at the start. It's really the magic comes in the months when it ages. And also, since the salt content is 4%, it also happens in a relatively quick time compared to a normal miso. We've aged our misos for a year, 2 years, and they turn into like a puck. They almost dry out completely, and that's because we start with a low water content so that the activity at 4% is semi-predictable, right?
So there's a reason why we're doing this. If you go to Japan, you'll taste very, very salty miso. Now, the Japanese, going way back in history, would have miso soup traditionally like 3 times a day. It was this like life-giving, warm manna that would feed you and nourish you, and it is an amazing food to eat and consume, especially diluted in a little bit of hot water. And that's because when you combine things like peas with molded barley, the nutritional content of both of those things together is higher than either of those things alone. Eating just raw peas is actually a bit hard to digest on your body. Eating just barley, not that interesting. And again, you won't get all the energy that it possibly contains. When you're fermenting foods, you're actually outsourcing the work of digestion, of the creation of all the enzymes that your body would have to produce to break these things down itself, to microbes out there in the world. Then you consume those things, the microbes themselves and the foods that they've left behind, and in that process, get something that's far more nutritious because the work has been done for you. Because all of these proteins are now liberated for what they are, amino acids that your body can then use to absorb and then build itself back up. These are the ideas of postbiotics. Now you hear like prebiotics and probiotics, eating living things or eating things that are good for the living things inside you, but no one ever talks about postbiotics. And postbiotics are all of the things that fermentation actually produces when you ferment. It is the sugar that's left behind after koji grows in a grain of barley. It is the amino acids like glutamate that your body finds so delicious because it's the poster child for all the other amino acids that it can use to build its raw materials with. And that's what taste actually is. Taste as like an evolutionary concept is fascinating because it's your body's way, just like going back to those ancient bacteria, tasting the world and sensing what was around them before the idea or concept of eyeballs was ever a thing. Taste is your body's way of understanding what part of the world to turn into itself. And all of your senses aid in your own survival. You have eyes so you don't walk off cliffs or into traffic. You have ears so you can hear an axe murderer sneaking up behind you on a creaky floor. You have smell to smell food, but also to smell things like fire of a burning attic or your basement or a gas leak. And these are all things that aid in your body making its way through the world successfully. Taste is fascinating because it's your body's way of figuring out what to make itself with. When you taste bitter compounds in the stalk of a poisonous tree, you spit it out and know that, yes, that tastes bad because it's poisonous. And it's only poisonous to you. It only tastes bad to you because, well, through the scope of evolution's lens, one of your ancestors or relatives died at the hands of that very chemical, and one of your ancestors that survived to actually produce the lineage that produced you had a unique variant gene that could recognize that chemical with an incentive attached to it, a negative incentive at that, an adverse incentive. When you taste all of the products of fermentation, you understand it is delicious because it is that heightened level of food. It is food that is extra good for you. It is the raw components that make up all of the proteins that you make your body with liberated and ready for you to build yourself with. And that's why fermentation is a fascinating thing to wrap your head around, because you understand it as life as like a very amazing and basic but also complex level. All life is transformation. Creation, destruction are just two sides of the same coin. Building things up in a certain way is just half of the picture if you understand that you have to break them down first. And when your body understands that something's been broken down for it in a friendly manner by something that isn't harmful in any way, alarm bells go off and you understand these things to be delicious. The search for that flavor, the creation of miso in this way, is how Renee and the team back in those test kitchens in the early days learned to use the very paltry pantry that sat at Noma's disposal with all these Scandinavian ingredients and multiply them many times over with the dimensionality of flavor that fermentation could provide. If first they only had, in a season, 250 ingredients to work with, they could turn to fermentation and all the different processes at their disposal to turn those 250 into 1,000. If each one of those things could find its own way to be fermented, yes, you could pickle a berry. Yes, you could juice it and turn it into alcohol and then use acetobacter to turn it into vinegar. Yes, you could dehydrate it and throw it into a miso so that you had a berry-flavored miso. Yes, you could preserve it by turning it into a garum or a fish sauce. Or you see the recombinatorial power that comes through fermentation. It's not just a subset of cuisine or gastronomy. It is, honest to goodness, as large, if not larger, than the world of gastronomy itself. That recombinatorial power that fermentation provides by mixing just, like you see here, analogous ingredients in ways that lead to tasteful outcomes is what makes fermentation as thrilling as it is to explore. Traditionally, things like soy sauce, things like vinegar, things like miso have had to be kept as very strict recipes and very strict guidelines so that they could be produced at all. Before there was Wikipedia, or before there was Google, you only had what your grandfather told you. Fermentation processes were always done in the homesteads of peasants and food producers at a very local level. And the fact that every pocket of Italy has its own special sausage for whatever village it comes from, whether that's Genoa or Calabrese, the fact that every town in Japan has its own type of miso, whether that's hacho or saikyo, speaks to this tradition. And yes, there is variance, but there's variance in all pockets of life. But still, the transference of DNA in organisms is as much about fidelity as it is about difference. And the same thing is true for things like fermented products like miso. It's as much about fidelity as it is about the possibility for change. And when Noma employs fermentation, we do it knowing that we can change things, that we can seek out flavors by looking to old techniques, by turning to tradition, breaking down those processes via reduction, pulling them apart, saying, what enzymes are at play? What molecules are in the mix? What byproducts would be produced? And then transfer that, kind of flip it over on its head to everything else in the possible, like to everything else at our disposal that could possibly be applied to. So we've made miso from a lot more than just peas and barley. We've made miso from,
I mean, anything you could think of, honestly, that is rich in protein, we've made miso out of it, honestly. Everything from seeds to
poppy seeds,
roots
sesame
that have a lot
seeds,
of protein,
valeriac, parsnip,
really dense meat pastes,
corn,
man, I mean, it goes on and on. Nixtamalized things.
fava beans.
Yeah, it just goes on and on. Byproducts, like whenever we press oil from nuts and we harvest all the oil out, we'll get all of the leftover chips that come from it, and then we'll make that into miso.
Yep.
Yeah, and even some really unconventional things.
Yep, we just did samphire and salicorn. So that's just the base product. That's just the legume. That's the thing that you're adding the koji to. But what happens when you grow koji on not just barley or rice, but buckwheat or wheat or
Vegetables.
or vegetables or corn?
Bread.
And then you start to see how all these things form lists that can be cross-referenced and form a web of just possibility that is really, 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 really big.
Yes.
That's all we do at the restaurant. Fill in this chart. 95% of it never sees the light of day. But the 5% that actually succeeds, that one connection where we said, oh, fava beans grown with Aspergillus soya and toasted wheat is amazing. That was what's in the Eppendorf tube, if you tasted that already. But that's a soy sauce that we made from an experiment that we probably tried 25 different variations on top of things that we'd done before to even get to that 25
Yeah, in one day, we made 25 different varieties
of soy sauce. Using different legumes, using different grains,
Yes.
and ending up at the conclusion 6 months down the line that this was the winner. And then that's what you're tasting right now. It's not soy sauce at all, because it's not made from soy. It's made from fava beans, and it's made from toasted rice, actually.
Yes, it was heavily toasted rice.
And for some reason, that magical combination, more than the red lentils and Oland wheat, more than the soybeans and toasted barley, worked extremely well. But that's what we do in the laboratory. We explore what I like to call flavor space. Flavor space is analogous to me what phase space is to an astronomer. When they're tracking the orbits of the planets around the sun and realizing that it's just slightly chaotic, that for every trip, the gravity of Neptune means that, Earth is moving in a slightly different orbit. And if you were to actually take a slice of that, you would have this beautiful pattern of never repeating trails. As a fermenter, as an exploratory fermenter, as someone who looks to tradition to understand the processes and then apply them to everything that history ignored, you're exploring flavor space. You're exploring those minute variations that just the chaos of existence leads to difference with when it comes to either fava beans or soybeans or peas or lupins. It doesn't matter, but they'll all turn out the same. If they're different at the outset, they'll be different in their outcome. That applies to making a batch of miso and all of the different control points that you could actually have that go into this. What was the ratio of peas to barley? How much salt did you use? How much pressure was there? How much salt did you add? What was the water activity level? Every control point in every fermentation process will lead to a different fermentation outcome. And then even when you're trying to do things exactly fungibly and make things exactly the same time and time again, we can attest things will go wrong. But it's not wrong. That's not the right word to use. They will go different. But again, that's the most beautiful part, that variation can spring up when you least expect it and lead to something new. The fact that there is Hacho miso and Aka miso and Okasan miso in Japan is a testament to this, except it just happened over the course of 1,500 years. The fact that there is balsamic vinegar and white wine vinegar and red wine vinegar in Europe is a testament to all these minute variations that led to different outcomes that people kept as traditions thereafter. But if you're not trying to keep things traditional, you're free to just do whatever you want. And that's the beauty of fermentation to us. There is this kind of second wave that Sandor started. Like, he definitely put Noma on its track by just illuminating the world and writing that tome and cataloging things and saying, this is what's out there. And it's almost as if he left crumbs to say, now run with it. And that's exactly what we tried to do with this book. This is less a book about recipes, and it is more a book about processes. It's less about particularities, and it's more about possibilities. Because if you read between the lines, you understand all of this just by reading about this. And that's exactly what Noma does. And it's really interesting that Rene wanted to write this book at all. I remember the day that he barged into the lab and said, David, we got to write a book. And I'm like, okay, I have to finish this first. We need it for service. And when we finally got time to sit down about it, he said, I want to publish everything because there's this moment brewing. There's this zeitgeist happening, and people are tuning in to the fact that fermentation has amazing power and possibilities, and we need to be the ones to actually let people know how it goes down. When I started cooking in restaurants, going back 15 years for chefs that worked for Marco Pierre White or Gordon Ramsay, they would keep the recipes in a lockbox under their desk, and you wouldn't get to see them unless you were the sous chef and actually had to put the special on that night. That's how the world of cuisine used to work. And it's really interesting that it's more about sharing and more about the transference of knowledge, because at the end of the day, I think Rene understands that this chart is way too big for us to ever fill in on our own. So by publishing all of our ideas, by publishing all of the discoveries, by running an Instagram account that is just basically our log of experiments, whether they're failures or successes, you spur on the field and you get to just have more toys to play with. And everyone does. And it's, of course, a beautiful thing. And that's why I find Noma to be a really cool place to work, because we're encouraged to explore, and we're encouraged to make mistakes, and we're encouraged to go down roads not knowing where they'll lead, but do so with the belief that we'll find something, good or bad. Whether the experiment works or doesn't, you learn something along the way anyway. I'm just going to, could I go back to the slideshow? Talk to you about the Nordic Food Lab a little bit right here. But this was the houseboat. So that project that Renee started, that was on here. And that was the first inklings. That was the beginning of all of these experiments that were the first forays into food preservation. This was maybe one of the first garums. Garum is a fermented sauce of fish that was invented by the Carthaginians going back over 2,000 years in northern Africa. They would harvest fish from the Mediterranean Sea, slice it up, mix it with salt to get the water activity level so low that nothing would grow, and then let all of the fish's own enzymes digest the protein itself, doing the same thing that we talked about when we were talking about miso, releasing free amino acids that you taste as umami-rich. It's the same thing that you have when you have Thai fish sauce, and you add that to a curry. And that's a great way to understand it. If you're ever cooking a curry at home, a coconut curry, and you have all of your spices, yeah, it's great. But it is missing that magic something until you add that few dashes of liquid MSG, because that's really what it is. All natural, though, which is nice. We use garums a lot at the restaurant. And again, that's by employing Koji's own enzymes, all of its proteases, to break down things that are much different from fish. Fish sauce tastes pungent. If you spill it on yourself, your date's probably ruined. But those proteolytic enzymes exist in both animals and in the fungus itself. If you swap out the guts of a fish for koji, you end up with a much cleaner tasting fermented product. And that's what we've done at Restaurant Noma. Again, synthesis via reduction. Break down a process, look at the pieces in play, and then build it back up as something new. And you end up with something new. So we've made fish sauces. Again, this was the Nordic Food Lab going back, this must have been 7 or 8 years ago. But we've made fish sauces from, or equivalents to fish sauce, from everything from squirrels,
Sea stars,
to beavers, sea cucumbers, meat like beef or chicken, reindeer, wild boar, pork, razor clams. Bear. That was the worst thing we've ever tasted. That did not go right.
But now we know.
Here is that same vat of fish sauce. You could see all of the bones of the fish just left behind because the proteins have been completely kind of chewed up by the enzymes of that molded barley. And that liquid is extremely potent. A few drops go a long way. And these were huge discoveries. No, it's true. These were huge discoveries for Restaurant Noma because all of a sudden you could have a primarily vegetarian cuisine that had all of the flavor and impact of like a full plate of food with meat and starch and vegetables just by using seasonings and kind of flipping the role of meat and vegetables on its head. Suddenly vegetables didn't have to be the accoutrement that just served to kind of temper what you were chewing in your mouth and acting as the texture. They were actually the starring role, but you were as satisfied as eating a full plate of sliced sirloin because you could have this taste of meat kind of permeating through your samphire mixed with watercress emulsion. More experiments there. But, and don't take this the wrong way, everyone at a university, academia and bureaucracy got in the way of the Nordic Food Lab's existence. At some point, there was like a board of directors in a chair, and they're like, why is this at a restaurant? This doesn't make any sense that it's on a houseboat. It should be in a university. And they actually took it away from Restaurant Noma. Even though it was something that Renee had created, it ended up kind of being divorced. And so it moved its physical location to Copenhagen University, and surprise, surprise, just sparks don't fly when you're 2 kilometers apart. Renee not ever wants to settle or stopped pushing, said, screw it, we'll build our own lab. And this was 2014. This was the second iteration of, well, I guess the first iteration of the fermentation lab, because by this point, fermentation had become Noma's main focus of research. And even though the lab did other things, primarily, it was always about fermentation. It was the most useful tool to the restaurant. So the fermentation lab was built by Ariel Johnson, who was a PhD graduate from UC Davis in flavor chemistry, and Lars Williams, who I mentioned before. It was built out of shipping containers as cheaply as possible and equipped with 7 rooms that were basically environments for every type of fermentation that Noma practiced. And that was the fermentation of vinegar, the fermentation of koji, misos and soy sauces, garums and black fruits and vegetables at very high temperatures, and so on.
Incubators.
And incubators, yeah. This is a very much younger version of Jason, but this was our kitchen.
Whoa.
This was our office, and it was amazing because at the time that it was built in 2014, this was 5 years ago, there was nothing like this in a restaurant on Earth. I mean, yes, there's Japanese fermenters that make their own rice wine vinegars and have rooms for their koji and things like this, but to have a laboratory just dedicated to every possible avenue of fermentation was unheard of. And the fact that Rene was willing to spend money to do it was also insane, and I'm sure his investors weren't too pleased. But he's a guy who believes in doing things for the sake of doing them, for the sake that they'll feed not back just into the restaurant, but into the world at large, which eventually it did. This is us making elderberry vinegar, and again, this is like three or 4 years ago. This vinegar is making its appearance on Noma's menu for the first time in 2 weeks. That's kind of cool. This is our old miso aging chamber, and you can see all the different varieties of things that we would ever produce. Now, Noma was built originally in a funny building that used to be an old whaling house. And as the restaurant grew and got more accolades and won awards and became the world's best restaurant, so did the restaurant. It eventually added a kitchen upstairs where we could do all the heavy lifting in production. It eventually built an office. And to get upstairs, you actually had to go outside. And that's romantic until you have to do it running up with a private dining room's worth of grilled greens in the middle of December, slipping on ice. Our dry storage was a building that was like 100 meters away down the street. Noma is DIY or die. It's do it any way you can until you can. And it wasn't the easiest place to work for this reason. Rene kind of knew this, and he also knew that he was getting a bit stagnant in his creativity. And that's when he decided to tear it all down and build a new restaurant in its stay. We did that 2 years ago, and in early 2018, we moved to an old bunker outside of the Christiania district of Copenhagen. This is Lab 3.0 that Jason and I built together in the new restaurant. This is the new team since Jason left. We have Diego from Peru and Kevin from Canada. It is a diverse mix in the restaurant, and 2 interns alongside us as well. And we have a lot of other fun technologies beyond just fermentation. Like I said, we are a laboratory. We're just called the fermentation lab because it's our main bag. But it is also up to me to keep the test kitchen entertained. There is the fermentation lab, but Noma also has a test kitchen that actually works with, it's Rene and 6 sous chefs, and they're actually the ones making the plates of food that end up on Noma's menu. They're the ones that we hand off interesting new products to and say, try this, or work with this, or this could be that thing that you need. So we're kind of like a bespoke research facility, tailoring to their needs, but also entertaining ourselves. Because at the end of the day, the guys in the test kitchen who are just trying to figure out how to perfectly grill duck breasts over Bitochen charcoal aren't reading scientific journals about what flavor molecules are responsible for Meadowsweet's almond flavor. That's up to guys like us. So Rene gives us full reign to just entertain ourselves, and so long as we get all our mise en place done at the end of the day and keep that innovation rolling, we're allowed to do what we do. So here, to the left, you see our cryogenic freezer. To the right, you see a supercritical fluid extractor. To the left, you see, it's hiding behind that speed rack, but there's a centrifuge and a rotovap, and our rational oven that we're in a research contract with. And it is an amazing space that kind of eschews all of the problems that we have if you ever tried to build a kitchen out of a shipping container. There are numerous problems. But it really is the dream. These are our fermentation chambers. This is our garum room, where we make all of those garums in the heat to actually get a roasted flavor added to them. And by doing that, we get to work with a little bit less salt. And this is our miso aging chamber, where we have all of our projects going. Anything you see in glass is just an experiment where we're trying things out for the first time. And should it be a success and the test kitchen like it, we'll ramp up to production to as much as 400 kilos of something a year. But it is an amazing place where we get to just explore for the sake of exploring. And just trying things out because we can. And this is just a paltry sample of all the things we've ever produced. I think I can end this. I'll just let you see, because my laptop's hooked up, our Evernote.
Oh,
but this is where we keep all of our documentation. And you can see just the sheer amount of tests that we go through. To make everything that we make. And there are like hundreds and hundreds and hundreds of experiments that we've gone through.
This is like only this year, I think.
Each one of these isn't just one experiment. I mean, for the game Boudin Noir that we were trying to make this past year is actually probably 20 different trials until we settled on something. All the different ways we could combine all the fermented products with blood pudding to make a tasty something that was eventually presented to the test kitchen and a final tasting with René Redzepi, where he said, "No thanks." But you understand just what we fill our days with when you look at something like this and see all of the things we try just to stay busy, just to entertain ourselves. And you might think that, well, isn't that kind of depressing that you work so hard for no thanks? But we don't see what we do as all for naught, because we look at it like knowledge creation. We look at it like explorers and say, okay, this door didn't lead to something, but it's worth knowing that that shouldn't be explored in the future. By publishing these results and actually sharing all these ideas with the public, you end up kind of enriching the world of gastronomy at all, I would say, and, I don't know, pushing the field forward.
Absolutely. And with our work as well, you know?
So I do have another slideshow to show you, which is just some of the actual projects that we've gone through in the lab and publish online. So this is our supercritical fluid extractor. This came about by, again, me actually going to a conference for cellular agriculture and talking with someone who'd seen me mess around with a rotovap, and he's like, oh, have you tried SFE? And I was like, what's that? And he's like, it's this amazing process of extraction where you use carbon dioxide to pull out organic compounds. And a year later, cross-pollination is an amazing thing. I was in contact with 2 perfume factories in the south of France, and they, excited by the idea, invited us down to work with this machine in their laboratories. And these are some of the biggest flavorists on Earth who are responsible for all the things you eat, who are responsible for all the things that you might spritz on yourself in the morning or roll under your armpits. Flavors and scents are everywhere. And this technology is one that they employ more than others to actually get things out without adulterating them in any way. So supercritical fluid extraction, I'll just draw this real quick, is a supercritical fluid is a fluid that is as diffuse as a gas but as dense as a liquid. And that's kind of a counterintuitive thing to think about. But that is absolutely true. If you were to evacuate all the gas in this room and open a balloon, all those molecules of air would expand and go and fill out at a very low pressure every corner of the room. If you were to take that same amount of volume, but have water and pour it out, it would spill out onto the floor and try and spread as much as it could, but you wouldn't be able to compress it very much. Above 32 degrees, 32.5 actually, and 74 bar, carbon dioxide enters a supercritical state. And that's the state that I just talked about, where it is as diffuse as a gas, but as dense as a fluid. And in that state, it is an extremely good solvent. It will strip whatever molecule you want into its stream, and should you revert that state back by dropping pressure or temperature, the CO2 returns to being an inert gas, and you get nothing but the product you want back at the end. It's really complex machinery, and it breaks down all the time. And I think no restaurant on earth has one of these in its lab, but it also makes some of the most potent, unadulterated, natural flavors you could ever imagine. We can probably only make enough of one product for a season, which is 3 months long using this machinery. The pilot unit that we have in the lab is probably about $150,000 US, but it's on loan to us from a company that thinks we're crazy enough to deserve it. But it is an amazing way to incorporate the essence of something into something else. We're using it on the current season with meadowsweet, an amazing herb that grows in the Nordics that has the flavor of almonds and marzipan, and incorporating it into a dessert that wouldn't be possible any other way. So we do get to kind of mess around with the states of matter by using technology like this. This was an extract of moss, and this one a petrichor, where we just went out into a field and asked our foragers to bring back the earth. We pumped it into the machine and ended up with something that tasted like kind of rolling around in wet leaves on a nice fall day, and actually used it to season a dish last game season of mushroom broth and lichen. So you had to drink this broth through the moss, your nose was buried in it, and all you could smell was a wet forest floor. And if you want to talk about moments and bringing people into a time and place, there's no more impressive way to do it than this. We also have our centrifuge, which is extremely useful, and it's basically put into use for hours a day, every day. And it allows us to get all of the waxes and esters and cells of chlorophyll out of things like sorrel juice, which you see here. You basically concentrate plant matter by spinning it at 4,000 Gs, and you end up with a clear liquid, but also something that's hyper-concentrated that you can then fold through a paste and apply to just one mouthful. We have our ultrasonic homogenizer as well, which we use to take normally immiscible liquids, like oil and fruit juice, blast with about 10 kilohertz worth of sound waves through a titanium rod, and end up with something that's the texture of milk. So in the way that molecular gastronomy came into fruition in the mid-90s up until the early 2000s, that was always about some sort, I don't want to say gimmick, but it was about a trick that was unexpected, about some sort of manipulation of a flavor for the sake of manipulation of a flavor or texture. A little like a huh moment. The way that Noma employs technology, whether it's biological or mechanical, it's always about improving something in a way you would never notice but couldn't explain. And when you taste the sauces in the restaurant's dining room, and you have something that's completely emulsified that shouldn't be, it coats your mouth in a different way. It doesn't dawn on you how that came about, but it completely affects the way you experience your meal. And that's what Renee tasks us with. This is our rotovap that we use for vacuum distillation. Another way of making essences, either trapped in vinegar or in alcohol that we can then season things on the menu with. And we also turn to kind of strange biological technologies as well that would normally not be employed in restaurants to really try and push the boundaries of what we can do in different directions. So fig leaves were a fun example of something that we tried to mess around with this year. Denmark is an amazing sector for biotech innovation, and there's awesome companies like Christian Hansen or Novozymes that brew using a lot of the same microbes that we talked about today, like designer kits of enzymes that can break down everything from feathers to bone matter to normally inedible leaves. So in a quest to turn fig leaves into something that was as pleasant to eat as spinach, we got an enzyme complex that was actually modeled after the digestive tract of snails. So by adding just a little bit of this and then going through, again, the scientific process with rigor, doing a test that was effectively 75 different tests, cooking things in water baths at slightly different incremental temperatures, you end up with something that dissolves on your tongue and has all of the flavor of a fig leaf in the end. This is it under a microscope, looking at the actually dissolved cell walls of plant matter, and seeing the protoplast. Is that the right term? That sits in its stay. I think that's what it's called, a protoplast. This is us messing around with duck feathers, a waste product from the game season when we had to pluck all the ducks ourselves. Again, using an enzyme complex to try and turn it into an edible food. This is something that's actually applied in the dog food sector. I know that sounds strange, but yes, chicken feathers are sometimes enzymatically decomposed to end up as filler in dog food. I mean, at the end of the day, it is organic matter that can be made sterile, that is full of amino acids and nutrients, and if you have a way of extracting that, you can end up with a usable product afterwards. Was this the greatest thing we've ever tasted? No. But we tried it.
Yeah.
It was inoffensive, but it wasn't delicious.
Like chicken broth and socks, kind of?
Yeah. You didn't want to spit it out, but you wouldn't ask for more.
Yeah.
This is us doing the lacto-fermented plums en masse. And this year, no lying, we fermented 750 kilos of plums. Like going to farms and being like, and getting the fruit every day, being like, is this the perfect ripeness? Is the perfect ripeness? And then being like, yes, give us 150 kilos. And then that's our job for the day. Lining them all up, IQFing them so that we can use all this stuff in the seasons where this fruit isn't available. Noma applies the basic concepts of fermentation, of preservation, of making something last in a season where it normally wouldn't be available, but taking it to the nth degree. So that we have 3 Michelin star fermented fruit in the dead of winter to make that menu brighten. Renee has definitely learned from that first mistake of the opening year, and it is now our job to exact those ideas every day. You want to talk about piquia?
You talk about piquia, actually, because that's your baby.
So this is a yeast that is not the common yeast that you find in a can of beer or your wine. This is something called Piquia cluveri. Actually, you would find this in wine.
You would.
And it grows in New Zealand in vats of Sauvignon Blanc. And New Zealand Sauvignon Blancs are well known for being extremely fruity in ways that Sauvignon Blancs grown in France or produced in France are absolutely not. Christian Hansen, a biotech firm that works in Denmark or is based in Denmark and is one of actually the largest on earth, invited us to see their headquarters just because, again, cross-pollination is a fascinating thing. And in their demonstration to us, they talked to us about their idea of making non-alcoholic beer better than it had ever been made before. Normally, non-alcoholic beer is just alcoholic beer with the ethanol removed via evaporation or other chemical processes. But if they could get all of the flavors of a non-alcoholic beer without ever having to produce alcohol, you end up with a much better quality beer. Piquia cluveri is a yeast that is responsible for producing all of the fruity esters from molecules of glucose and maltose, but has no fermentation pathway in the traditional sense, and it doesn't produce alcohol. It's also not an anaerobic organism, it's an aerobic one. It's actually considered a fault or a contamination yeast in a lot of other processes, but here, if you look at it for what it is or what it can do, it is something absolutely fascinating. So once we got our hands on this strain, we applied it to everything we possibly could. That included the juice of berries, the syrups made with pine cones, fruits.
Koji water.
Yeah, exactly. Worts made with koji.
Miso water, all kinds of things.
Everything. And we ended up with an amazing array of products that were Nordic ingredients, but transformed with tropical flavors of banana and passion fruit.
Grape, chewing gum.
Grape chewing gum. Yeah, seriously. Yeah. And it is, again, an all-natural process that gives us another kind of sector on that chart of recombinatorial power to make new flavors with, in a way that no fermentation process I'd ever read about had before. So that's a fascinating one. We also work with a lot of traditional processes and just do minor tweaks using things that are available in our region. This is us making batarga, but burying it in our miso, ending up with exactly fish eggs that you can shave over whatever product you desire that taste like nothing you would see before. And in that same vein, the fermentation of things that Koji would not normally work with is something that we explored a lot this season. Renee, bolstered by the success of the book, is like, all right, double down on fermentation. And this past vegetable season, we put out 3 dishes of moldy vegetables that Jason actually pioneered working with to figure out how to do. Because Koji doesn't normally like growing on vegetables. It likes grains. But if you know how to work with it and know how to baby it, you can make dishes like this.
Yeah. I found that it really increases like the volatiles of the actual ingredients. So like with the asparagus, for instance, it wasn't necessarily like a long fermentation process. It just went through incubation. But the actual asparagus aroma and the flavor was amplified significantly. It was nice.
This is that same aspergillus under a microscope that we have in the lab. And again, working with things like spirulina, you can make miso from things from the ocean, end up with very, very different flavors. Green going in, blue coming out. Ferran Adria recently ate at Noma, and he got to taste this. He was just hanging out with me for a little bit, and he's like, I think his mind melted out of his ears when he saw this. He's like, why is it blue? Nothing is blue. The fava bean miso that you guys tasted, these were the initial experiments, us doing it for the very first times. This is what our miso room looked like, just jars and jars and jars and jars of different varieties of soy sauces to end up with that final delicious flavor. But that's what we do. We try, this was pickled eggs for this coming game season. We try everything at our possible disposal. Run tests of dozens of variations of things to end up at one final answer that might be something that's never been created before or the best version of something that's existed for a long time, like a pickled egg. This is that egg pickled in that elderberry vinegar that you saw me making 3 years ago that's been aging in Brookladdick whiskey barrels for this whole time, reducing slowly. That is the best mouthful of food you will taste in a long time. And we had to can 5,500 partridge eggs in 2 weeks during the high season so that we could have them for the last 3 months of this year. Random experiment. And also working with things that you normally wouldn't eat. This is us trying to extract tomato leaves this past summer, trying to get that amazing aroma that you get when you brush up against a tomato plant, and just using every technology at our disposal. So no, it's not always about combining flavors and seeing which one's the best. Sometimes it's about combining technologies and seeing what produces the best flavor. So we tried all different sorts of extraction, sonication, vacuum filtration, the SFE unit, to end up at was eventually a rotovapped distillate with its base product added back in that was that exact flavor that you wanted. Is it used anywhere? No. But hey, at least we know we tried. The preservation of things that you wouldn't normally eat falls on our shoulders as well. This is the nixtamalization of pine cones to render them less bitter and more tender. That's them at the beginning, and this is them coming out. And the nixtamalization of other fruits that you wouldn't normally run processes with. Mugaritz did a lot of work with this over the past 10 years, but it is really fun to combine their work with our work, and you end up with black fruits and vegetables that don't degrade after being kept in the heat for 6 months, but stay firm. And you could take a bite out of this apple, but it also tastes like a tarte tatin. And also looking to other places in the world of, or in the natural world for inspiration and fermentation. This is one of your projects,
Yeah.
Chris?
Yes, absolutely. I mean, just like we look for koji to break down the proteins and the starches in these ingredients to make a miso, in the same way that this grain is capable of producing amylase, we were looking more internally. And also, we looked to the animal kingdom for inspiration. And I found that I went down this rabbit hole of extracting pancreas enzymes from pigs and then fermenting the meat of the pig with the pancreas enzymes. My first trials were trying to get the pure enzymes extracted out with all kinds of technologies. But David was like, hey, Jay, you're stressing out a lot over here trying to do this extract. Why don't you just try blending it into the actual meat, the whole pancreas? So we actually found that just taking a little bit of this waste product that people eat in certain communities and adding it to a kilo of meat can completely liquefy it into pure umami in under 5 hours. It doesn't taste the same as a garum because the process doesn't really involve a lot of caramelization. But at the same time, it tastes like an incredible bouillon that you would have like in a soup, like a store soup, something like that. Incredibly impactful.
That was the last slide. Good timing.
Okay, are you sure that was
Yep.
the last one?
Yeah.
Wow.
Oh, that was. There's no more slides. We're done.